So in 9.3, we are going to be talking about the effects of pH on microbial growth. So remember that pH here, pH is the measure of hydrogen ions in a solution. Measure of hydrogen ions in a solution. And recall that the pH scale goes from 1 to 14, with 7 being neutral, with anything above 7, so 7.1 to 14, being alkaline, and anything below 7 being acidic, meaning that anything below 7 has a high hydrogen ion concentration, and anything above 7 has a low hydrogen ion concentration. So most of our organisms are neurophiles. Most of our organisms enjoy being in or need to be in a neutral environment, so somewhere around 7, and we'll talk more about that in just a moment. But this is because of our effects, or the effects that pH has on macromolecules. So this is the same within our bodies as with all organisms. Our bodies and other organisms all work best at specific pH ranges. And most things need close to neutral pH, and this is because of the macromolecules. So the pH effects on macromolecules. So this is, for example, the hydrogen bonds in DNA. Hydrogen bonds in DNA. Those are going to be disrupted. Those will break depending on the type of pH. So we already have hydrogen bonds, and depending on whether or not we're more alkaline or whether or not we're more acidic, then we're going to have that affect hydrogen bonds. So if it affects hydrogen bonds, it actually affects hydrogen bonds overall, but if it affects hydrogen bonds in DNA, for example, then we're going to start to be breaking apart our DNA. Our DNA is going to be falling apart to pieces, and that is not conducive to life. Also, the lipids are going to hydrolyze. So lipids will hydrolyze. And remember that our lipids, our lipids are made up of long carbon-hydrogen chains. So if we're talking about our hydrogen bonds being affected, remember that we have some molecule here depending on the type of lipid we're talking about. And then all of these have hydrogens coming off of them. These are the carbons here, if we remember the way that we draw macromolecules. These are our carbons, and then hydrogens are coming off of each of these carbons with these long fatty acid tails. And so if we're disrupting these hydrogen bonds, then we're having a serious effect on lipids as well. Another way that things are affected are hydrogen gradients are disrupted. And so hydrogen gradients are important, of course, at the end of the ETS, the electron transport system, where we have that proton motive force. So remember, proton motive force meaning that as part of our exchanging of electrons from protein to protein in the plasma membrane, this is embedded in the plasma membrane in the mitochondria, for example, we're pumping hydrogen ions into that inner membrane. And because we're pumping these hydrogen ions across the plasma membrane here, then at the end here, those hydrogen ions, this concentration going back down is what's going to allow us to make ATP. And so if we are adjusting or disrupting our hydrogen ions, if we have a serious increase in hydrogen ions, then we have a disruption of this difference in hydrogen ion concentration between the inner and the outer. Or if we have a serious decrease in hydrogen ions, then we're going to be decreasing all of the hydrogen ions. Therefore, we're going to have a serious problem with making ATP. So our hydrogen ion gradients would be disrupted. And then last but not least, our proteins can denature. And this is a serious concern as well, because most of our enzymes are made up primarily of proteins. So when we have an increase in hydrogen ions, those are going to interact with those R groups, or those different individual amino acid groups in our long protein chains. So remember that our proteins are made up of individual amino acids. Those individual amino acids have different charges on them. And an increase or a decrease in hydrogen ions is going to affect very greatly the charges on those amino acids. And then that's going to unravel our proteins. If we unravel our proteins, then those enzymes are no longer effective. If the enzymes are no longer working or effective or even exist, then we don't have things like transcription and translation. And then we don't have, and even so much as the actual structure of the cells and structure of the organism will fall apart because the proteins themselves fall apart. So pH is a huge concern. And so we need to make sure that as we're cultivating or inoculating things and trying to grow microorganisms, that we have them in their correct pH range. Now, I've been saying correct pH range, and that's because there's actually a minimum growth pH. And what this is, as its name implies, is the lowest pH an organism can tolerate. Conversely, we have our maximum growth pH, which of course is then the highest pH an organism can tolerate. And then an organism has its optimum growth pH. And of course, this would be the most favorable condition in regards to pH, of course. So as we are talking about different terms here, moving on, we're talking about kind of this optimum growth pH for each organism, and we always try to keep it in its optimum growth pH if we're trying to cultivate it in a laboratory. Or even when we're talking about trying to isolate an organism from a person, we can actually use these different pH ranges in order to identify different organisms. And that's because oftentimes, as I've mentioned previously, organisms are neutrophiles. So many organisms are neutrophiles, particularly those when we're talking about a medical laboratory or research facility. If we are studying something that is going to be a pathogen for humans, for example, or other animals, typically we're talking about neutrophiles, and that's because human bodies and other animal bodies are generally neutral, around 7 in pH, and so those organisms that are going to be pathogenic to those types of organisms are also going to be an optimal range of about 7. So really, the optimal range that we're talking about here, though, is a pH from 5 to 9. And it really depends on the organism. So some neutrophiles, for example, are E. coli. So that grows on and in our bodies. Oftentimes, it's not a problem. It's just part of our natural microbiome. But it grows in and on us because we're at about 7 to 7.5, depending. So that is the optimum growth pH, but it can be between pH 5 and 9, depending on the organism. Also, Staphylococcus species, those are also neutrophiles. Salmonella is a neutrophile. It can go pretty low, down to that pH range of 5 region, so it can get into something pretty acidic, so that's why we can see it in spoiled things, for example. But then others, like E. coli, doesn't typically do very well at a pH that's closer to that 5. It's better closer to that 7 and maybe up a little bit higher. So those are termed neutrophiles. Then we have acidophiles. And acidophiles, as you can probably guess, like things to be acidic. And their optimal pH is going to be less than 5.55. So there's a little bit of overlap between like neutrophiles, acidophiles kind of thing, but generally it's below 5 or below 5.5, the text says, so below 5. And so some examples are sulfolobus, and lactobacillus. And you may be familiar with lactobacillus just kind of in a general sense, because lactobacillus is what we often find in probiotics. It's often what we find in things like yogurt, Greek yogurt, regular yogurt. and curdled things are due to Lactobacillus. And that's because in our microbiome, in our intestines, we have a lot of Lactobacillus species, lots of Lactobacillus genus in our intestines, and that keeps us healthy. We work together. And as you can imagine, if we have our nice acidic stomach that's breaking things down, we're going to have something that can withstand acidity, even as we get into the intestines. So the intestines are not as acidic as the stomach, but they do still have a lower pH than our kind of 7.5-ish of the rest of the body. Our blood is generally between 7.35 and 7.45, so that's why I'm saying general. So it's important that our blood stays within that 7.35 to 7.45. So some organisms aren't able to live in the blood because that's what the blood range is. So for example, acidophiles will not do well in the blood. They need to have more acidic environment, but they'll do well in areas like the stomach or the intestines, for example. Now, how can these organisms do this? They have developed adaptations over time. And so some of the adaptations, for example, their proteins are different than proteins of neutrophils. Their proteins have an increased number of negative surface charges. So negative surface charges. And then that stabilizes them at that lower pH. So again, it's a way that the body has adjusted. So these proteins, remember, are so important that they can start to unravel. So the organisms here, like Lactobacillus, for example, actually have differences in their proteins to have more of a negative surface charge so that the increase in hydrogen ions isn't going to affect them as easily as it would affect a neutrophil. They also have adapted to have pumps. So pumps that actually eject hydrogen ions out of the cell. So they don't want there to be that much hydrogen inside of them, so they just have pumps throughout the organism, throughout the cell, that are just pumping excess hydrogens out of their little cell. And then also they have a change in the composition of their membrane phospholipids. So a change in composition of membrane phospholipids. And so again, our phospholipids are made up of our phosphate group, our glycerol backbone, and then we have our fatty acid tails. And remember, we already discussed how our lipids can be affected by pH. So they actually have a change in the composition so that the hydrogen ions don't affect them as much. Therefore, their plasma membranes can remain intact. All right, so those are the acidophiles and how the acidophiles have adjusted to have optimal pH less than 5. Next, we're going to take a look at Alcala Files. So our Alcala Files, of course, like things more alkaline. So the optimal pH for an Alcala File is going to be anywhere between 8 and 10.5. Really anything above 8. But we haven't really found organisms that live above anything higher than 10.5. And so we say 8 to 10.5 are the Alcala Files. So for example, an Alcala File is Vibrio Cholera. So Vibrio Cholera, I spelled that wrong. Cholera. So Vibrio Cholera is something that can affect humans, but that's if the pH is going to be amenable to them. Their optimal pH is the 8 to 10.5. So they have to be able to take hold in an area that has a higher pH. So some of the adaptations that we see here are going to be lipid and protein structure changes. So again, lipid and protein structure. So for example, for the proteins, instead of having an increased negative surface charge, instead we're going to see an increased positive surface charge. Then they also have mechanisms that maintain the proton motive force. So they have changed. So again, remember, proton motive force is when we're in the electron transport chain and we are moving the electrons along the chain and we're pumping hydrogen ions out of that inner membrane, or from one side of the membrane to the other side of the membrane. And then that allows them to come back in and make ATP. So Alcala Files, since they are in a situation where there's such low hydrogen ion concentration, they've actually adjusted their proton motive forces. And so some of them, for example, use sodium ion gradients instead of hydrogen ion gradients because sodium ions are more available than hydrogen ions. So they've adjusted their mechanisms to maintain proton motive force. So, for example, they'll use a sodium ion gradient instead. And they have a higher isoelectric point. So a higher isoelectric point. And that is due to the more basic amino acids that they have in their proteins. So again, they're going to change protein structure. So not only is there going to be an increased number of positive surface charge on the proteins themselves, but then they're also going to be utilizing more basic amino acids because then they're going to have an easier time existing in this alkaline environment. Okay, so to summarize this, of course, we need organisms to be in their optimal growth environment, or that's how we study them, would be in their optimal growth environment. But then that also is what allows us to kind of narrow things down. So if somebody has an illness and we determine that it's in the blood, for example, we can go in and we know that we don't have to test out organisms that are acidophiles or organisms that are alkalophiles, because if they're in the blood and the human's blood is between that 7.35 and 7.45 range, then what we're looking for are neutrophiles. And so we can study things at a neutral pH. Now, if we find something is in the stomach, then perhaps what we need to do is we need to shift our study to something that's less than 5 pH. And so then when we're trying to cultivate the organism, we would put it in an environment that is a less than 5 pH. This also helps us when we're not talking about human health, but when we're talking about studying the world at large. So, for example, the soda lakes are going to have very basic, it's a very, alkaline environment. And so we're going to be looking at alkalophiles, and we're going to study them in an alkaline environment versus the acidic ponds and puddles, for example, in Yellowstone, that are going to have acidophiles, and we'll be studying acidophiles and then making sure that we have that pH range. So in general, we have talked a lot about the pH range, and you should be familiar with these terms: the minimum growth pH, the maximum growth pH, the optimum growth pH, and then you should understand what neutrophiles are, acidophiles, and alkalophiles, but you should also know, so not only the ranges that we have here, but you should also know the adaptations, so how it is that they can live in a more acidic environment, how it is that they can live in a more alkaline environment.